Maybe I was a fool to hang on to someone that would never change. Maybe I was a fool, never let on just how much my heart was strained. Maybe I...
All right, let's talk about the nervous system. Divisions of the nervous system. We have the central nervous system, often referred to as CNS, and then the peripheral nervous system, often referred to as PNS. The central nervous system is composed of the brain and the spinal cord, and the peripheral nervous system consists of cord-like nerves that link the central nervous system to the rest of the body. So all of our peripheral nerves that we often talk about when providing local anesthesia or blocks in general, we're talking about the peripheral nervous system. Functions of the nervous system, we've got sensory functions, integrating functions, and motor functions. The nervous system senses changes from within the body or from outside the body, and it conveys this information to the spinal cord and the brain. In the brain and spinal cord, the sensory information is received, analyzed, stored, and integrated to produce a response. A motor response instructs the body to do something, such as contract a muscle or cause a gland to secrete its products. The study of the nervous system and its parts, of course, is called neurology, and that's a really big topic of investigation. So the basic unit of the nervous system that demonstrates nervous system functions, such as response to stimuli or sending out or taking in impulses, is called the neuron. So this is a picture of a neuron. So it's the basic functioning unit of the nervous system. And these are, of course, present in both the CNS and the PNS. And we'll go through each little aspect of the neuron in a few minutes here. A few interesting facts. Neurons can't survive without oxygen for long at all. If we think about it, cardiopulmonary resuscitation, so CPR, has to be started immediately. Immediately. So as soon as that heart stops, we need to start CPR immediately, because the nerve cells start to die immediately without oxygen. Technically, the heart could start beating again, as sometimes we often get the heart to beat again by giving epinephrine, a whole bunch of different drugs, unfortunately. So we can get the heart to beat again quite often, but there could be serious brain damage once oxygen depletion has occurred, and that's because that nervous system tissue has died. It dies very quickly. They are like a skink. They can regenerate. So looking at the neuron itself, they have the ability to regenerate. So if a piece of the neuron falls off, is torn away, whatever it might be, they can actually regenerate pieces. As long as the majority of their cell body is intact, then neurons have the unique ability to regenerate, kind of like a skink. When it loses its piece of its tail, it can regenerate a piece of its tail. That being said, shortly after an animal is born, the neurons lose their ability to reproduce, so they don't undergo mitosis shortly after the animal is born, but they can regenerate cell processes. So this lack of reproductive ability is why serious nervous system injuries, such as stroke and spinal cord damage, are often so debilitating, and they have long-lasting effects. But, interesting thing, emergent research is suggesting that it may be possible to actually turn on the neuron's reproductive ability. So this is something that they're looking into, which would have huge implications for individuals, both animals and humans, who have gone through fairly significant neurological deficit. So whether that be damage, spinal cord injury, whatever it might be, but there is a lot of potential there for those studies. Then last but not least, neurons are needy. They're very needy. They always have this entourage around them. So they have these things called glial cells that are present to support them, continuously support them in their daily activities. So going back to our image of the neuron itself, we'll go through each component part. The dendrite receives stimuli and impulses to conduct to the cell body. So it receives it from other neurons or from other cells, and it's conducted through the cell body, out through the axon, and of course down. The axons conduct nerve impulses away from the cell body toward another neuron or effector cell. So effector cell is a cell that does something when stimulated, such as a muscle or a gland cell. And then we have the myelin sheath, which is actually the cell membranes of specialized glial cells called oligodendrocytes in the brain and spinal cord, and Schwann cells in the nerves outside the brain and spinal cord. So that's the actual cell membranes of those cells. And then on this one in particular, this is a Schwann cell, and this is a cell in the peripheral nervous system that forms the myelin sheath around the neuron's axon. I'll talk a little bit about the myelin sheath, when we start talking about synapses and the action of the nerve itself. So in the peripheral nervous system, we call these we call them Schwann cells, and in the central nervous system, so the brain and spinal cord, they're called oligodendrocytes. And then we have the node of Ranvier. These are specialized, the node of Ranvier works together with the myelin sheath to increase the speed of conduction of impulses along the axon. So we'll talk about that a fair bit as well. And then lastly, we have the axon terminal, which is the portion of the neuron that releases the impulse to the adjoining neuron or cell. It's also known as the terminal bouton. And here's a nice little image of oligodendrocytes, which of course are the cells involved in the central nervous system along the brain and the spinal cord. And they essentially wrap the axons in nice little myelin sheath coating. Okay, so nice little myelin sheath sweaters. So the cranial nerves are the nerves of the PNS that originate directly from the brain. And the spinal nerves are those PNS nerves that emerge from the spinal cord itself. So looking at this cat here, right here, these are all the cranial nerves. So they come direct, they're not all really there, they're just sort of giving a brief description. But the cranial nerves, it's important to remember that they stem directly from the brain. And the rest of the peripheral nerves essentially at some point stem from the spinal cord. Looking at that, some nerves conduct electrical impulses from the periphery toward the CNS, and other nerves conduct impulses in the opposite direction, so from the CNS toward the periphery. These two functional types of nerves are called afferent nerves and efferent nerves. Afferent is to conduct nerve impulses toward the CNS. Okay, so toward the CNS for afferent nerves. Efferent nerves conduct nerve impulses away from the CNS. And you can see here we've got some efferent nerve descriptions there. Because afferent nerves conduct sensations from the sensory receptors in the skin and other locations in the body to the central nervous system, afferent nerves are often called sensory nerves. In contrast, efferent nerves conduct impulses from the central nervous system out toward muscles and other organs, because the efferent impulses are the ones that, among other things, cause skeletal muscle contraction and movement. The efferent nerves are often called motor nerves. Cranial and spinal nerves in the peripheral nervous system and nerve tracts, which are bundles of axons in the central nervous system, may carry nerve fibers that are sensory, motor, or a combination of both. So generally important to note that afferent nerves run toward the CNS, so toward the central nervous system, and they're also called sensory nerves because they're taking in the stimuli that's affecting the sensory cells and bringing it toward the central nervous system for perception. And then efferent nerves are taking those signals away from the central nervous system, and they're also called motor nerves because they often result in movement of the skeletal muscle. Memory trick, because there's a lot of things that we have to get to know in anatomy and physiology. For me, efferent nerves, in order to remember that they run away from the body, I think of this, F off. I know it's really rude, but F off is to go away, and that's efferent, away from the central nervous system. Inappropriate little memory trick. Don't write that on your tests. Okay, so then we get into autonomic versus somatic. So when an animal turns its head in response to its name being called by its owner, that is an example. It's a conscious, voluntary control of skeletal muscles. So that is an example of somatic nervous system function. because the action of that animal turning its head away was caused by voluntary initiation of efferent impulses. This function would be classified as somatic motor function. Somatic motor function. So somatic, anytime the somatic system is affected, we're talking about voluntary conscious effort. The other aspect, of course, is our autonomic. So impulses being any impulse that is automatically controlled by the body. So continuously beating heart, continuously reproducing cells, continuously hormone producing glands, etc. Any of those involuntary sort of subconscious or not conscious movements or processes are considered part of the autonomic system. So the part of the nervous system that controls and coordinates all of those automatic functions is the autonomic nervous system. And of course, sensory nerves and motor nerves can affect both the automatic and somatic, but sometimes, depending, one might weigh a little bit more so than the other. Such as an example, of course, motor nerves, they're taking in a cue, which is sensory, but then they're using their central nervous system to send out motor signals to create that somatic involuntary response. All right, so now we get into the trickier part of how do they work? How do these neurons actually function? How do they fire? What's it all about? So the cell membranes of neurons are electrically charged even at rest. That's called the resting state, and it describes how much they're positive on one side, negative on the other. Because ions generally, especially the ions that we're going to be talking about, sodium and potassium, they have various charges. In this case, they're both positively charged. Because them, as well as the chloride ions and the carboxylate ions that are sitting within the cell, also have a negative charge, within that we get the resting state, which is the level of electrical charge within the cell and outside the cell. The charge of the cell will vary depending on the phase of polarization. And the phases of polarization is exactly what we're going to get into now. So a resting cell, sodium doesn't naturally diffuse through the cell membrane. Sodium accumulates on the outside of the cell, and it maintains an overall positive charge on the outside of the cell membrane. On the other hand, potassium doesn't naturally diffuse out of the cell. So potassium sits in high volumes inside the cell. However, chloride, which is negatively charged, and carboxylate, also which is negatively charged, they also hang out inside the cell with potassium. It's like a little gang they've got. So that being said, because we have all tons of sodium, positive sodium outside the cell, and we have a fair bit of potassium inside the cell that's positive, and then we have some secret, which isn't shown on here, chloride and carboxylate, which are both negative inside the cell, it ends up causing a negative charge inside the cell and a positive charge outside the cell. This is called polarized. So this indicates that the cell is polarized. If we think about magnets, we think about polarized. They go to opposite ends, negative versus positive. The potential difference is more sodium outside the cell than potassium inside the cell. And of course, polarized because it has two distinct poles of ions on either side of the membrane. Now, most photos that you'll look up, most photos in general, they don't identify that there's chloride and carboxylate inside the cell, which really attributes to the negative charge. So always keep that in mind because I find resources can be tricky on that one. All right, then we have resting membrane potential, which is the electrical differences across the cell membrane, which equals a negative charge inside. Then we have the impulse, so the nerve impulse is coming through. It connects with the dendrite and enter the sodium and potassium channels. So we've got the nerve impulse coming through the dendrites. We've got sodium and potassium channels now opening up to accommodate for that nerve impulse and to get into action. So the impulse travels into the neuron from the dendrite. The sodium channels allow only for sodium to flow into the cell, and it maintains the potassium inside the cell currently. So this now is called depolarization. So what's happening, the influx of sodium comes into the cell because the little sodium channels have opened. The potassium is currently staying inside the cell, which means that with that added sodium, the inside of the cell becomes quite positive. Likewise, the outside of the cell still has sodium outside, so it's also positive. So that is depolarization. It means that we no longer have a positive and a negative. So the sodium, when we think about it, once this neuron, this electrical charge starts to come in through the cell, coming in through the dendrite, and those little sodium channels open, the sodium itself is encouraged to flow into the cell because A, they're drawn to the more negative charge of the inside of the cell, right? You take away that barrier and negative and positive are actually attracted to each other. And B, that old thing called the concentration gradient. There are now more sodium ions outside the membrane, so they actually want to distribute themselves inside the cell membrane in order to maintain homeostasis. This immediate change from the cell being negatively charged to positively charged is called an action potential, because the next step is, of course, repolarization. So repolarization, so what we had during depolarization originally was this little sodium gate was open and the potassium channel was closed. So that was allowing an influx of sodium into the cell. So now during repolarization, it's the opposite. The sodium channel or the sodium gate closes and the potassium gate opens. So what ends up happening is potassium escapes the cell. So potassium starts heading out of the cell. Again, because of that concentration gradient, it recognizes that there's more potassium inside the cell, so it wants to even things out. So this rush of potassium ions out of the neuron causes a swing back to the cell being negatively charged. Negatively charged inside the cell has been restored, but it's a little bit backwards. So now, again, if we think about it, so initial steps, that electrical impulse came down through the dendrite. It enters to the cell body, and then at the base of the cell body, right at the start of the axon, is where all this is happening. So essentially, the sodium channels all along the plasma membrane recognize that an impulse is coming through. They open up. They allow sodium inside the cell. It makes the inside of the cell positive. The body doesn't like that. So immediately, because we have positive inside the cell, positive outside the cell, immediately sodium channels close, potassium channels open, and it allows a whole bunch of potassium outside the cell. So if we think about it, it's the opposite of how we began. So now the potassium ions are outside the cell membrane, sodium ions are inside the cell membrane. So it's kind of good because we have negative on the inside, positive on the outside. So again, we're polarized, but we need to restore the balance of the sodium and potassium ions. So they need to be in their respective places. So what we end up acquiring is the sodium-potassium pump, which is an active process that causes redistribution of the sodium and potassium ions. So instead of just the sodium-potassium channels, which were passive, we now have this active mechanism whereby the sodium-potassium pump plucks out the sodium that's inside the cell, it sends it outside, and it plucks the potassium from outside the cell and brings it inside. So it just redistributes everything selectively to maintain its original state. All right, so recap. Resting state is the polarized state where sodium is outside the cell membrane, potassium is inside the cell membrane, is a general a general negative charge inside the cell thanks to that additional chloride and carboxylate, which are negatively charged. This is considered polarized. Then we have the depolarization phase where a nerve impulse comes through the cell, comes in through the dendrite, passes through the cell body. As it's passing through, the sodium gates will open and allow that influx of sodium to enter into the cell. That screws up the balance, and it now makes the inside of the cell very positive, as well as the outside of the cell is quite positive. So this is called depolarization, and this is the action potential. This is the phase at which the neuron is firing. Okay, depolarization is synonymous with the neuron firing. Also, this phase, when we have any of the channels opened, and also when the sodium and potassium are not distributed as they are in the resting state. This is called the refractory period. So during the refractory period, it's when the neuron is in the midst of depolarization process and it's actually unable to respond to another stimuli, which is really important because, of course, if we continuously had neurons firing while they're in the midst of repolarization, then we wouldn't get a very good system of neuron charges or of neuron activity in muscles. It would just be continuous and uncontrolled. So not ideal. So this is called the refractory period. And the neuron generally cannot accept another impulse. So then we have the beginning of repolarization. So those sodium channels close, potassium channels open, and potassium shifts to outside the cell. Okay, so now it's getting back to that negative on the insides and positive on the outside. Again, this is part of the refractory period. And then finally, final stages of repolarization. The sodium gate completely closes, potassium gate is closing. Sufficient outflow of potassium has reinstated the polarized effect in the cell, like I said before. So repolarization. And then, this is the final stage where the sodium-potassium pump just redistributes that sodium to the outside the cell and potassium to the inside of the cell. And remembering that the sodium-potassium pump is an active process and it requires the use of ATP from within the cell. So that was depolarization in a large nutshell. There's this thing called the depolarization threshold. So not all signals will be strong enough to actually cause the neuron to depolarize. Weak signals, so weak electrical signals that come from the brain and from the central nervous system and peripheral nervous system, they may cause just a little bit of sodium to enter into the cell. So they may trigger that sodium channel opening. Some of the sodium comes into the cell, but instead of flipping it entirely because it's just not weighted enough, so instead of flipping it into depolarization stage, the sodium pump kicks on and plucks out those sodium ions from inside the cell, and it kicks them outside the cell. So it's just this active process that kicks on and just plucks out the sodium ions that have come into the cell. It knows they don't belong. The neuron then goes back to the resting state. The impulse was not sent to the brain. The depolarization threshold is a point at which the stimulus is strong enough to cause depolarization. And it depends, because when we talk about cardiac muscles, the cardiac muscles in the sinoatrial node essentially to some point have the lowest level of depolarization threshold because they're the ones with the fastest contractility, so the fastest rate of contraction. And of course, they set the pace for all the other cells in the heart. So the conduction of nerve impulse, we have the conduction of action potential. This wave of depolarization happens. So once one little area on the plasma membrane starts firing, starts depolarizing, it sends this wave of depolarization all the way down the cell. So from the body of the cell, through the base of the cell, all the way down the axon of the cell. Okay, it's this big, huge wave. And when a neuron starts firing, it's all or nothing. There is no half firing, there's no weak firing. It's all or nothing. They go all in for their impulse firing. So just to confirm, they get an impulse sent through the dendrite, works its way down through the cell body, and then essentially at the base of the cell body is really where that depolarization wave starts. So we get depolarization through the cell membrane, and then of course that wave of depolarization runs all the way the length of the axon, and of course the goal is to end in the terminal boutons, or the axon terminals. So then we have these things called the myelin sheath and the Schwann cells, or the oligodendrocytes if it's in the central nervous system. And then, of course, the nodes of Ranvier. So the presence of a myelin sheath allows for nerve impulses to travel faster, or at least for that depolarization wave to happen faster. As the sodium channels are opening to cause depolarization, the myelin sheaths maintain the sodium ions within the limits of the membrane, so it actually avoids losing them to the space outside the cell. It essentially allows them to shift at the point of the nodes of Ranvier. Therefore, It's as if depolarization doesn't even have to happen all the way down the membrane, but it skips chunks, which is essentially covered areas of the myelin sheath. It skips those chunks by depolarizing at the nodes of Ranvier. Clinical application for this one that's fairly important. Individuals with multiple sclerosis have damaged sheaths, so have damaged myelin sheaths on their neurons. Really important. So the loss of normal muscle control and an inability to make coordinated movements, and sometimes even see properly, is the result. With a damaged myelin sheath, the nerves may still conduct the impulses, but their conduction is much slower, resulting in a loss of the rapid communication needed for complex integrated functions of the nervous system. So that's something to keep in mind when it comes to myelin sheaths. Clinical application. Local anesthetics are applied to superficial nerves, so in the peripheral nervous system. The presence of these local anesthetics, such as lidocaine, marcaine, bupivacaine, their presence blocks the sodium channels and thus prevents depolarization of the sensory pain nerve, which is called the nociceptor. So what's happening is it's as if they're plugging those little sodium channels. It stops them from opening to allow that sodium into the cell, into the neuron cells, or the neurons themselves. And then without sodium influx inside the cells can't depolarize. So that's how local anesthetic works. So we have synapses and boutons. Nerves are antisocial. They don't like to actually touch each other. Instead, there's a little gap, so little synapse between each of the nerves. The terminal portion of the axon is called the bouton, and the target cell or another neuron connect together via this synapse, so this little gap. An impulse is sent through, we've already identified that it's sent through the nerve, through the axon toward the terminal bouton, and there's a little vesicle of neurotransmitter that's released and diffused into the postsynaptic cell, into the postsynaptic cell. The postsynaptic membrane is covered in receptors, and the receptors are very selective. So each receptor is very selective to certain types of neurotransmitters. So we'll look at that a little bit closer. So we've got a nerve impulse coming through the axon, works its way to the terminal axon into this terminal bouton. And in the terminal bouton, we have all these little vesicles, so these little sacs that contain neurotransmitters. The postsynaptic cell, this is the dendrite of the postsynaptic cell, and in their cell membrane on the dendrites, they have little receptors. So the receptors only accept little neurotransmitters that fit them perfectly. So in this case, we can pretend that this is acetylcholine being released by this terminal bouton, and the postsynaptic cell on its membrane has little receptors that are very specific to recognize and engage in action when acetylcholine is released. So in this case, we've got perfect little circular acetylcholine being released by the synaptic space, by the presynaptic membrane. Those circular little pieces of neurotransmitter acetylcholine fit perfectly with the neurotransmitter receptors. Okay, and once they fit perfectly, like a missing puzzle piece, then it can have an impact on the adjoining nerve or cell. Neurotransmitters, they can be either excitatory, which encourage depolarization by creating an influx of sodium into the cell to help it move toward that threshold, or inhibitory, so they can hyperpolarize the cells to maintain negative charge on the inside of the cell. So if they hyperpolarize, then the chances of depolarization are decreased. Perfect example, benzodiazepines such as Valium or diazepam, same thing. They enhance the effect of GABA, and GABA is the main inhibitory neurotransmitter. So GABA, when it is stimulated, when GABA enters into the body, what actually happens, GABA's effect opens chloride channels inside the neurons. So because of GABA, more chloride enters the cell itself. And of course, we know that chloride has a negative charge, so GABA essentially causes the cell to become hyperpolarized, which means that it won't fire. This has a tranquilizer effect on the animal because it's actually inhibiting the natural effect of depolarization of that cell. So it's inhibiting the action of that cell. Common neurotransmitters, we have acetylcholine, which can be both inhibitory and excitatory in the parasympathetic nervous system. Actually, where is it? Yeah, parasympathetic nervous system. Acetylcholine has an inhibitory effect on the heart. So it actually increases bradycardia, which means it slows the heart rate down. But all other skeletal muscles in the body, acetylcholine has an excitatory effect. When acetylcholine is released to a muscle cell, it causes contraction. Norepinephrine causes arousal. It's part of the fight or flight sympathetic nervous system response. Epinephrine is released by the adrenal gland and it acts as a hormone as part of the fight or flight response. Dopamine is involved in autonomic functions and muscle control. And GABA, which I talked about a minute ago, gamma aminobutyric acid, has an inhibitory effect so that it creates sort of tranquilizer effect on the body. Norepinephrine, epinephrine, and dopamine are called catecholamines. And dopamine is often people with decreased number of functioning dopamine neurons show muscle tremors and the uneven gait commonly associated with Parkinson's disease. So they suspect that dopamine and a lack of dopamine receptors and dopamine functioning dopamine neurons actually can be contributed to some degree of Parkinson's disease. Okay, so now our neurotransmitters have been released into this postsynaptic space, and they're just floating around like crazy. So don't we think that if the neuron has dumped a whole bunch of neurotransmitter into the synaptic space, won't it just keep floating around and keep getting absorbed by that postsynaptic membrane? Won't it just keep stimulating the postsynaptic receptors? Nope, it won't. What ends up happening is the body recycles neurotransmitters because they're so valuable to the body. So there's this thing, every neurotransmitter essentially has an enzyme that's associated with it to break it down and recycle it into its component parts. One that we commonly talk about is acetylcholinesterase, and that in particular is the enzyme that essentially is sent out to break down acetylcholine to stop that postsynaptic neuron or postsynaptic cell from continuously firing. Clinical application. Organophosphate toxicity is when, so essentially the organophosphates, which are things like pyrethrins, which is, quote, medication contained in a lot of tick prevention and in some flea prevention. Essentially, in cats, it destroys or deactivates acetylcholinesterase, and it's unable to stop the muscle action. So in normal muscle action, acetylcholine is released. We'll just go back to here. Acetylcholine, normal muscle function, the neuron releases acetylcholine from the terminal bouton, connects with the receptors in the muscle cell, and of course, acetylcholine causes the skeletal muscle to contract and contract and contract. Normally, in a normal situation, acetylcholinesterase would then be released and it would break down that extra acetylcholine. But when cats are given some of these tick preventions that contain pyrethrins, which are called organophosphates, what ends up happening is that the organophosphates destroy or deactivate that acetylcholinesterase. So what ends up happening is overstimulation of the acetylcholine receptors. So what you end up getting is, of course, a very shaky kitten who's continuously having little muscle tremors, and they can be quite severe. And we know that with continued muscle tremors, we know that muscles produce heat. So of course these cats can overheat very easily. If an owner puts on this medication and then they very quickly realize that they put on the wrong medication for their cat, it's recommended that they bathe it immediately and then bring the cat in for follow-up care because it will need to have those tremors stopped. Really, really, really scary stuff for cats. So things like Heart's Care, the flea and tick drops, contains pyrethrins. This is not safe for cats. And likewise, Bayer and Vantix, they do a pretty good job of warning clients that it is not for cats. This is also pyrethrin, and we do not ever apply it to cats. So it's good to know how organophosphate toxicity actually occurs and damages cats and their muscles, their synapses. Okay, So now we're going to talk about the brain. It's so complicated. It's so complicated that we as humans don't even know how the brain works in its entirety. But that's okay. We don't need to know that either. We need to know how areas of the brain are linked to different behaviors and abnormalities. So we're just going to talk about some very basic parts of the brain and what they might be responsible for in the body. So we've got central nervous system is the brain and spinal cord. In the brain, we have the cerebrum, cerebellum, diencephalon, and the brainstem. For each of these, we'll talk about them in very brief detail. Very brief detail. So the cerebrum, very important part. Cerebrum is the area of the brain responsible for higher order behaviors, such as learning, intelligence, and awareness. As you can imagine, humans have a very well-developed cerebrum. Moving on. In the cerebrum, we have the gyri or the gyrus. Folds, this is the folds in the cerebral hemispheres. So the folds all throughout the cerebral hemispheres. The fissures are the deep grooves separating the gyri. The sulci, or sulcus in singular terms, is the shallow grooves separating the gyri. It divides the cerebral hemispheres into the lobes themselves. The longitudinal fissure is the prominent groove that divides the cerebrum into the right and the left cerebral hemispheres. The cerebellum we talked about a little bit in detail. So the cerebellum is located just caudal to the cerebrum. Here it is, just hanging out. Hey, little buddy. And it's the area of the brain responsible for coordinated movement, for balance, for posture, and for complex reflexes. Do you guys recall cerebellar hypoplasia? Cerebellar hypoplasia, of course, is an underdevelopment of the cerebellum, and it happens to kittens whose mothers had panleukopenia during gestation period. And of course, they end up looking like drunken sailors. They have uncoordinated, very ataxic and large movements. So the diencephalon is kind of a big deal. It's the passageway between the brainstem and the cerebrum. The structures associated with the diencephalon are the thalamus, which acts as a relay station for regulating sensory inputs to the cerebrum, and the hypothalamus, which is the interface between the nervous system and the endocrine system, and the pituitary, which is the endocrine master gland. So the pituitary is essentially the most important part of the endocrine system overall, and it has implications for all of the endocrine glands. And we'll talk about that in more detail when we talk about the endocrine glands. And then we have the brainstem, which is the connection between the rest of the brain and the spinal cord. It's composed of the medulla oblongata. It's probably one of the best terms in the world, medulla oblongata, the pons, and the midbrain. This is the area of the brain responsible for basic support functions of the body. So it's one of the very basic parts of the brain that was developed. And many of the cranial nerves originate from this area in particular within the brain. The meninges are connective tissue layers that surround the brain and spinal cord. They contain blood vessels, fluid, and fat. They supply nutrients and oxygen to the superficial tissues of the brain and spinal cord. They also provide some cushioning and distribution of nutrients for the central nervous system. Inflammation of the meninges is called meningitis, and this is probably a term that you've heard before. Most often, meningitis, which is inflammation of the meninges, is caused by viral and bacterial contamination. And we'll look at the next slide at the various layers of the meninges. So we've got 3 layers of the meninges. We have the dura mater, and it's tough and fibrous. That's the outermost layer. We have the arachnoid layer, which is delicate and spiderweb-like. And then we have the pia mater, which is very thin and lies directly on the surface of the brain and spinal cord. Cerebrospinal fluid, this is the fluid between the layers of the meninges and in the canals and ventricles inside the brain and central canal of the spinal cord. This provides cushioning function, and it may play a role in the regulation of autonomic functions such as respiration and vomiting. Cerebral spinal fluid we often will collect. If the doctor suspects that there might be meningitis present, then typically a CNS fluid or cerebral spinal fluid will be tapped and taken and tested for any viral or bacterial contaminants. So now we're going to move on and talk briefly about this blood-brain barrier. So the blood-brain barrier is very important, and it separates the capillaries in the brain from the nervous tissue. The capillary walls, so capillaries of course are the smallest of all vessels. They're about 1 cell layer thick. The capillary walls in the brain have no fenestrations, so they don't have any little holes in them. And they're covered by cell membranes of glial cells. Normally, capillaries are fenestrated all throughout, and they're about a cell thick, and they are the location of most nutrient exchange or oxygen and carbon dioxide exchange, and they do that through their little fenestrations. Okay, pop. The lack of fenestrations in these little capillaries in the brain prevents many drugs, proteins, ions, and other molecules from readily passing into the blood, from the blood into the brain. So it acts as a slight barrier to prevent some of those larger ions from getting into the brain itself. So instead of a lot of drugs having direct access to the brain via circulation, instead they have to get rerouted and metabolized and then be taken up by the brain in a different form. So we'll look at the blood-brain barrier a little bit here. So here is a normal capillary epithelial. So epithelial cells that line most capillaries in the body have gap junctions between each cell to allow for the passage of molecules to and from the bloodstream. The head itself has many, many, many capillaries. That's why heads always bleed when you cut them by accident. I mean, like if you bump your head or something, nothing weird. Capillaries in the brain are lined with special epithelial cells that have tight junctions. So here are the capillaries in the brain, part of the blood-brain barrier, have tight junctions, and you can see, having a look at the circle of epithelial cells here, where you have small gaps in between them. Here, you have nice tight junctions. So the blood-brain barrier, because of its really, really tight junctions, it stops most molecules from crossing this barrier, but it does let some through, so it's semi-permeable. If a molecule is fat-soluble, it can diffuse through, or if a molecule is just really, really, really tiny, it can use diffusion to cross the barrier. This is how psychoactive drugs have essentially an immediate effect on the brain. Other molecules are actively transported from the blood to the brain through special transport methods. Example, of course, is glucose. The main fuel of the brain is received through the blood-brain barrier. So it's good to know about the blood-brain barrier, and especially how it prevents some drugs and other ions from entering the brain right through circulation. Okay, that it has to get rerouted through the body, through metabolism, to get sent back up to the brain in a different format. It will come up a lot more in pharmacology. So then cranial nerves, we talked about a little bit. There are 12 pairs of nerves in the peripheral nervous system that originate directly from the brain. They're numbered in Roman numerals from 1 to 12, and each nerve may contain axons of motor neurons, axons of sensory neurons, or of course both. Really, really, really important factor in facial surgery. Anytime the doctor is working with the base of the ears, so around the tympanic bulla, essentially, tympanic membrane, or on the face itself, they have to be very aware of where these facial nerves originate from. The cranial nerves, I mean. So for us, doesn't have too much impact. Of course, if we are looking at dental blocks, then we might have some clinical application there. But otherwise, we as RVTs are not performing facial surgery. And then we have the spinal cord, which is made up of the medulla, which is the central part of the spinal cord. It's composed of gray matter, so not containing a myelin sheath. White matter contains a myelin sheath, and it looks fatty, or it has that fatty myelin sheath on the outside. It tends to look white on macroscopic examination. The central canal is the center of the medulla. The cortex is the outer part of the spinal cord. It's made up of white matter mostly, and it surrounds the gray matter. Okay? Looking at we could go on a lot more about the spinal cord, but in this case we won't. But it's important to know just all about the spinal cord in regard to how it's protected within the vertebrae, and of course where during an epidural, where we give that epidural medication in order to cause a generalized local block. So the autonomic nervous system, we talked about a little bit previously, so it controls the automatic functions at the subconscious level. Within the autonomic nervous system, we have the sympathetic nervous system, which are nerves that emerge from the thoracic and lumbar vertebral regions. And then we have the parasympathetic nervous system, where nerves emerge from the brain and the sacral vertebrae, or vertebral regions. Things to know about this, of course, is the difference between the sympathetic and parasympathetic nervous system. And we have to keep in mind that it's all about the R. So the sympathetic nervous system is the nervous system that's responsible for fight or flight. So it's responsible for hyping up the animal to prepare to fend itself in a fight, in a battle, or to run away at really high speed. The parasympathetic nervous system then kicks on afterward and is all about rest and recover. And we'll look at a little chart that describes the functions of each in a minute. So then here, of course, we're looking at the effects, where it affects the body in the sympathetic and parasympathetic nervous system, and exactly how it affects the body. This is straight from your textbook. And I just love this. I'm going to give a little shout out to the awkwardyeti.com. It's a fabulous comic that often focuses on all the body systems that we talk about in these lectures. Okay, here's that handy dandy chart. So the effects of the sympathetic and parasympathetic nervous systems. Sympathetic nervous system will increase the heart rate, increase the force of heart contractions. It dilates the bronchioles to allow more air to flow in. It dilates the diameter of the pupil. It decreases GI secretions and motility and blood flow. It decreases the diameter of skin blood vessels, and it increases the diameter of muscle blood vessels, and it decreases the blood vessels to the kidney. So essentially what it's doing, it's pumping the heck out of the heart, and it's rerouting a lot of that blood from the unimportant areas, such as the skin, the superficial extremities, or the superficial aspects of extremities, and it's pumping it all toward the heart and the lungs. Parasympathetic does the opposite, decreases heart rate, no significant effect on the force of heart contraction. It constricts the pupils, it decreases the diameter of the bronchioles to go back to normal, it increases gastrointestinal motility, secretions, and blood flow, so it brings it back down to normal, has no significant effect on the diameter of blood vessels, muscle blood vessels, or blood vessels to the kidney. So fight or flight for the sympathetic, and rest and recover for parasympathetic. We'll talk a bit more about the neurotransmitters and their receptors. So the primary neurotransmitter of the sympathetic nervous system is norepinephrine. Norepinephrine is an adrenergic neuron, which means that it comes from the adrenal gland. And those are neurons that release norepinephrine. Norepinephrine is a neurotransmitter secreted by adrenergic neurons. That's what I meant to say. Epinephrine and norepinephrine are also released from the adrenal medulla. So looking at this image, very important, if you can picture it. So this is a zombie, just to clarify, it's a zombie. And this young lady is getting chased by the zombie. She's spilling her milkshake all over. So in this case, would it be parasympathetic or sympathetic nervous system that's activated? Well, for her, actually for both, because it's fight or flight, it's the sympathetic nervous system that's kicked on. All right, looking at these neurotransmitters, so blood vessels in the skin, the GI tract, and the skeletal muscles have adrenergic, so catecholamine receptors, to epinephrine or norepinephrine. Alpha-1 adrenergic receptors cause vasoconstriction of the skin, GI tract, and kidney. Beta-1 adrenergic receptors increase heart rate and force of contraction. And beta-2 adrenergic receptors cause bronchodilation. And we talk about these a lot when we talk about cardiac medications in pharmacology. Primary neurotransmitter of the parasympathetic nervous system is actually acetylcholine. So when it comes to the parasympathetic nervous system, acetylcholine actually has inhibitory effects on the heart, so it slows down the heart. Cholinergic neurons are neurons that release acetylcholine. Don't forget too that acetylcholine has an excitement effect on skeletal muscles, but an inhibitory effect on the heart. This will all come back to haunt you in pharmacology. And that's about it. So again, zombie, fight or flight. We're talking about the sympathetic nervous system.
For thinking that I might be worth your time. Maybe I was a fool for dreaming